So today we're going to begin Chapter 3, talking about prokaryotic cell structure and function. So first off, what do we mean when we say the word prokaryote? Well, historically, prokaryote has always lumped together what we now know to be two distinct groups of organisms, the bacteria and the archaea. But previously they were lumped together because at the time, this is back in 1962, it wasn't really clear that there were two distinct organisms. So Stanier and Van Niel first described prokaryotes based on three characteristics. One, they have no membrane-bound nucleus, which is unlike eukaryotes, which do have a membrane-bound nucleus. Prokaryotes have no organelles, unlike eukaryotes, which have quite a few organelles. And they also describe prokaryotes as having no cytoskeleton. And generally speaking, what we can still say today is that prokaryotes differ from eukaryotes in terms of size and simplicity. Generally, prokaryotes are a lot smaller than eukaryotes, and they're also generally a lot more simple because they don't have the organelles eukaryotes do. So even though we have this classification initially, since 1962, we've now found that planktomycetes, which is a prokaryote bacteria, actually does enclose its genetic material in a membrane. So now we have an example of a prokaryote that does have something similar to a membrane-bound nucleus. And we've also discovered cytoskeletons present in both bacteria and an archaea. So it's not really quite cut and dry what is a prokaryote anymore. This has led to what's known as the prokaryotic controversy, where we're not really sure what we describe as a prokaryote. But anyways, we'll still go on and talk more about prokaryote structure based on these the general rule, which is these three characteristics described by Stanier and Van. So let's talk a little about bacteria more so, because we're going to focus more on them than archaea this semester. So talk about bacteria cell shape and arrangement, basic types of structure. So in terms of cell shape, the most common two types of shapes are either cocci, which is a fancy word for circles or spheres, and rods. And so there are less common shapes that can be possible. We'll go over a couple of those in a second. And then in terms of the arrangement, so when we look at bacteria under the microscope, which we will in the lab, we're going to see not just the shape, but also there are certain arrangements. So some bacteria may be arranged in chains, where you'll have lots of rods or cocci together connected to each other. They may have no particular arrangement. There's also kinds of really interesting arrangements that we'll talk more about. So what determines the arrangement of bacteria cells is the plane of division. So when two cells divide, what plane is a division on? Is there only one plane of division, so they all divide to create chains, or are there multiple planes of division? And then also, is there separation after division? So after they've divided, do the cells fully separate from each other and go their separate ways? Or do the cells remain together, which would change what the arrangement appears like under a microscope? Alright, so let's look at the cocci shape first. Again, mentioned fancy word for circle. So here we have our cocci, or spheres. And there's several different types of arrangement we can have these cocci. We have the diplococci, which when you have two pairs of spheres together, so if you had just two circular objects connected to each other, two circular cells, that would be a diplococci. We have streptococci, which you can see this example over here on the top, the strep agalactiae, cocci in chains. And we have what are known as staphylococci, or clusters of cocci, which are shown down here at the bottom. So that's when you have not so much change, but actually clusters of cocci. They kind of look like clusters of grapes here. You can see the gram stain where they're showing purple. Look like clusters of grapes under the microscope. And the last is what's known as tetrads, or squares. And this isn't actually a square shape, because bacteria don't come in square shape, but it's basically when you get four cocci arranged to make what appears to be a square. So two on top, two on bottom, and it appears as a square. And then we have the rods, which is also known as bacilli. So that's the plural version. Singular version, a single rod, is sometimes you may see it referred to as a bacillus. Careful note here, make sure you don't get this confused with bacillus, in italics here, which is a genus of bacteria. So rods are sometimes known as bacillus, but there is a whole group of organisms called bacillus, which are rod-shaped, so that makes sense. But don't get those two terms confused with each other, because that is different. So a couple types of rods. You can see here on the top where we have bacillus. This is B. megatherium here. These are rods arranged in chains. And you can see the difference in rods compared to cocci. We don't have circular shapes now. Now we have what are more kind of brick-shaped organisms. We can also have what's known as coccobacilli, which are basically very short rods that sometimes appear as cocci under the microscope. So it can be difficult to distinguish between the two because they're just very, very short rods that can look like cocci. Vibrios are a type of rod, so that's a comma-shaped rod you see in the bottom right here. We have this vibrio-shaped. This is Vibrio cholerae, which causes cholera. Very comma-shaped Vibrio. So you ever see a rod that has kind of a curve in it like a comma, that's a Vibrio. And then other bacteria shapes. So we have what are known as spirillums, which is basically this rigid helix. If you look at this top organism here, the C. jejuni, that's Campylobacter jejuni, which causes a lot of foodborne illnesses. We have this spiral-shaped spirillum here, so it's kind of got this interesting spiral shape, and this is a very rigid structure. This is in contrast to a spirochete, which you see down here below that we have Leptospira interrogans, which is a spirochete, which is somewhat more flexible. So you see these don't really fit into the rod or coccyx shape. They have their own classification. These are either spirillums or spirochetes. And the only difference between those is whether they're rigid or flexible. And then we have what are known as filamentous or mycelium, so filamentous organisms. Down here you see Streptomyces. It's kind of difficult to see here, but the long kind of filamental parts here, not so much the circular parts, but these long filament parts, that's Streptomyces, which is a very filamentous bacterium. It produces these nice filaments. Streptomyces produces a lot of the antibiotics that we've discovered. It's a huge producer of antibiotics, so kind of an important organism that's living out there in the soil. And then we have what are known as pleomorphic organisms. So these are organisms that don't really have a particular shape, or they have multiple shapes and something that's not easily described. So here's two examples of that. This hyphomicrobium here, you can see how it actually has this kind of hyphae, which we generally only see on fungus, but this is a bacterium. And we have this bud, kind of hyphal shape, so there's not a lot of one individual shape here. And then on the right we have M. stipitatus, which is a fruiting body that's found on a particular type of bacterium. So let's talk about the size of bacteria and prokaryotes. And so I mentioned that the size of prokaryotes generally is much smaller than that of eukaryotes, but that being said, there are still many different sizes possible for prokaryotes and bacteria. So the smallest bacteria known is Mycoplasma, which is about 0.3 micrometers in diameter. And so 0.3 micrometers, if you look at this, it's not shown on this chart here, but if you look, we have this in nanometers. 0.3 micrometers is equivalent to 300 nanometers. So if you look at Mycoplasma, it's about the same size as this pox virus down here, and that's an animal virus. So we actually have an example of a bacteria that is around the same size as a virus, which is kind of impressive because we always think of viruses as being really, really small and a lot smaller than bacteria, but there are extremes in every organism, and bacteria also do have the extremely small, and pox virus is an extremely large virus. So there's no cut and dry indication on when the size of a virus stops and a bacteria starts. There is some overlap in the possibilities. Average size bacteria, an average size rod, would be something like E. coli, which you can see here, which is about anywhere from 1 micrometer in width to about 2 to 6 micrometers in length. And so E. coli is, as you can see, smaller than a red blood cell, larger than a Streptococcus cocci shaped bacteria. And then we have very large bacteria. So one example is Epulopicium fischelsonii. There's a really neat picture in your book that shows this organism next to a eukaryotic cell. So Epulopicium fischelsonii is about 600 micrometers in width by 80 micrometers in length. So it's a huge organism for the bacterial world. It's actually about the size, similar in size to what actually would be like a grain of sand. So it's you can actually see it visibly without a microscope. All right, let's talk about the organization of a bacterial cell. So if you just look at this picture here that's showing a general schematic of the bacterial cell, you can see that it is, in general, much more simple than that of a eukaryotic cell. And all of you have seen a eukaryotic cell at some point, which has organelles coming everywhere. This is a lot more simple. So let's kind of start from the outside and just walk through this. We have the flagellum, which we'll talk more about for Monday's lecture, and the flagellum is basically used for motility. So it's how certain organisms that are motile are able to get around. We have fimbriae, which are used for, can be used for motility, but are mainly more used for attachment, so that the organism can attach to certain surfaces. That can also play a role in pathogenicity. When a cell is infecting you, how it attaches to the cells in your body. A lot of organisms, particularly those that are pathogenic, will have what's known as a capsule. So this is a layer outside the bacterial portions of the bacterial cell wall and plasma membrane that is often composed of polysaccharides. So you can see it's the outermost layer. It's really sticky and can often allow a cell to either attach to certain surfaces, or it can help the cell evade the immune system because it's such a sticky layer. It's difficult for the immune system to actually grab a hold of. inside the capsule, if a bacterium has them, all bacteria are going to have a cell wall. So not everything has a capsule, but all bacteria, with maybe a couple exceptions, have a cell wall. This is one of the major things that distinguishes them from animal cells, which our cells don't have a cell wall. So we'll talk more about the cell wall in a minute here. Plasma membrane. So this is very similar to animal cells. The plasma membrane of bacteria serves many of the same purpose as it does for animal cells. So it's a selectively permeable barrier, which ensures that only compounds we want inside the cell are actually able to get inside the cell. Plasma membrane helps the bacteria interact with the environment, contains receptors that can detect and respond to the environment. Bacteria have some really neat ones that we'll talk about this semester. Also serves as transport system, so a lot of the proteins that allow nutrients to transport into the cell, and a lot of metabolic processes occur in the plasma membrane as well. Alright, so we'll briefly go through these that we'll talk more about later. We have some inclusion bodies in bacteria, which is kind of the form of storage they use. Just like our cells, bacteria have ribosomes, or theirs are slightly different, which are used for protein synthesis. And then instead of a nucleus, bacteria have what are known as a nucleoid, which is a region in the cell that contains the chromosomes, DNA, and all the proteins that go along with them. Alright, so let's talk about the bacterial cell envelope. And basically what the envelope is the plasma membrane, the cell wall, and anything that may be surrounding the outside, be it a capsule or any other component. So the fluid mosaic model of the plasma membrane in bacteria, you can see here, looks pretty similar to what we see in eukaryotic cells. There's not a ton of differences. Now, we have our lipid bilayer, we have some proteins that are inserted in between. A lipid bilayer is kind of similar in bacteria as it is to eukaryotes, although the types of lipids that compose it maybe will be different, but the bilayer, the structure is generally very similar. So we have peripheral proteins here. Just a reminder, those are the ones that don't actually extend through the membrane, but they attach to proteins that are in the membrane. So they're very loosely connected, and those can be more easily removed than our integral proteins, which are embedded in the membrane. And so integral proteins serve a lot of functions. One of the main one is going to be transport of nutrients, ions, and such across the membrane. And then we'll see here this hopanoid. That's something you likely haven't really seen before. So hopanoid is basically the bacterial's version of cholesterol. So we have cholesterol in our membranes. Bacteria don't have that, but they have what are known as hopanoids, which serve the same function. They help stabilize the membrane structure. Alright, so the cell wall, I mentioned all bacteria have them. So the cell wall is a rigid structure that's located just outside the plasma membrane, which of course you remember from that image. And the cell wall is made of a compound known as peptidoglycan, which is something not found inside our cells so much. So there are two types of cell walls. We have what's known as the gram-positive, this G+, and the gram-negative, G- cell wall. So I'm going to use the terms G+ and G- because gram is just way too long of a word for me to write out all the time. So the functions of the cell wall are mainly to maintain the shape of bacteria. So bacteria have the shape they have for a reason, so the shape needs to be maintained. And also protect the cell from osmotic lysis. So if a bacteria found itself in an environment where there was a sudden decrease in solutes, so like if you put a bacteria into DI water where there's nothing but water in there, a lot of cells, like our red blood cells, would quickly lyse. They would burst open because the water would rush into the cell and the cell would lyse. Having a rigid cell wall protects bacteria from this because they have this rigid structure that's going to prevent sudden lysis from sudden influx of water, and it's going to prevent the cell from bursting because it's really rigid and strong. The cell wall also protects the cells from toxic materials. So it's one future step in protection is also to prevent toxic materials from getting into the cell that could, especially toxic materials that could potentially diffuse across the plasma membrane. So it has to get to the cell wall first. And the cell wall can contribute to pathogenicity. This is in particular for gram-negative organisms where we have this problem where a portion of, it's not their cell wall, it's actually something attached to their cell wall, but actually can contribute to the pathogenicity of. So let's talk about the difference between gram-positive and gram-negative cells. So the main difference to keep in your head is that gram-positive cells at the top here have a really thick layer of peptidoglycan. So you can see in this upper right corner here, this peptidoglycan layer in gram-positive cells is very thick. Gram-negative cells, they don't have that. They have a much thinner layer of peptidoglycan. So if you look down at the bottom image here, in between the plasma membrane in pink and the outer membrane on the left, we have this really thin layer of peptidoglycan. So that's a gram-negative cell wall. It's much thinner. Gram-positive cells also have a very small periplasm. And so the periplasm is, if you look down at the gram-negative image, you can see it's actually the region in between the plasma membrane and the outside or the outer membrane of the cell. So gram-positive bacteria, they have a very thin periplasm. Theirs is basically just between the plasma membrane and the peptidoglycan layer. It's very, very small, so they don't have much periplasm. And gram-positive cells may contain what's known as teichoic acids. So we'll talk more about that in a region, but that's something very specific for gram-positive cells, is these teichoic acids. Gram-negative cells, on the other hand, they have a large periplasm, so it actually made their image. Their periplasm is all the way from the plasma membrane to the outer membrane. We have this periplasmic space here. It's much larger than in gram-positive cells. And like I mentioned, they have this outer membrane. So they have this whole other outer membrane layer of protection, which helps them. Since their cell wall is so thin, they need an outer layer of protection. So they have this outer membrane layer of protection that contains lipids. So it's a lot kind of like the plasma membrane. It has lipids, although it's a little different in the structure. Lipoproteins, so proteins attached to lipids, and also lipopolysaccharide, which is known as LPS. So this compound, which we'll talk more about, this is a compound that is very important for the gram-negative cells, and particularly those that cause infection, so those that are pathogenic. This LPS helps them establish pathogenicity, and it also has a big role in modulating the ability of the bacterium to evade the immune system. So this is a very, very important molecule. All right, so let's talk about this peptidoglycan. Since it's kind of new, it's very unique to bacteria. So peptidoglycan is basically a mesh-like polymer of identical subunits forming long strands together. Okay, what does that mean? So basically, to break it down for you, we're going to have this polymer. So we have two alternating sugars here. We have this N-acetylglucosamine, or NAG, which you can see in this darker orange color over here. And we also have that with N-acetylmuramic acid, or NAM, which is in the yellow here. So you're going to have alternating, this long chain that has alternations of NAG-NAM, NAG-NAM, NAG-NAM connected to each other over and over again. So this disaccharide repeated over and over again for a long chain. Connected to that, particularly connected to the NAM, we're going to have alternating chains of amino acids. So there's alternating L and D amino acids. So you need to go back to organic and remember what L and D was. And you don't have to really remember, just know that there's a difference in the structure between L and D amino acids. L amino acids are present in proteins, D amino acids are not. So you get this alternating chain of L and D amino acids that come off our NAM here. So we have, in this example, we have alanine, glutamic acid, diaminopimulic acid, alanine. So there's a long chain here that comes on. And that's going to be used for cross-linking our sugar chains together. So in this example here, here's our NAM and our NAG with the 4 chains of amino acids. And then this last chain here is actually linked to an amino acid that's connected to NAM-NAG of another polymer. So we're going to have 2 long chains of polymers of our sugars that are going to be linked together by cross-linking these peptides. And so the cross-linking, how it occurs, there's a variety in how this cross-linking can occur. So we can have just what you see here, this example where alanine directly connected, one amino acid directly connected to another. Or down here in B, you can see we also can have what's known as a peptide interbridge, where we can have glycines connected to each other, so more amino acids connected to each other, linking one of our chains to one of our peptide chains to another. So we can have this alternate. Basically, we're going to link these chains together. Now, if we expand outward and look at the 3-dimensional structure of peptidoglycan, we'll see that it's actually not just a straight chain. It actually forms a helical shape. So our NAM and NAG here, that polysaccharide backbone, if you look over here in figure 318, we have our alternating NAG and NAM in the same orange and yellow colors. That's actually going to curve over and form this helical shape itself. So we're going to have this helical shape of polysaccharides. Each NAM is going to have its peptide side chain coming off it. And then those are going to be connected together. And because there's multiple types of crosslinks possible, and again, the crosslinks are multiple. There are different types of crosslinks that can occur. And the reason that we're cross-linking these together is this is going to produce a lot of strength. So having this helical shape and then cross-linking one helix to another helix, we're going to get this really intense mesh polymer that's going to have a lot of strength, which is going to add to the strength of the cell wall. So down in this image here, this is actually a portion of peptidoglycan, a big meshwork crystal structure of peptidoglycan looking at by electron microscopy. And you can see, well, you can kind of see, it's not a great image, but you can see there's kind of this really intense meshwork overlapping of the different sugar backbones with each other. So it's really kind of this intense structure. And so in class, we're going to look more at the structure of peptidoglycan and the cell wall, distinguishing between the gram-positive and the gram-negative cell wall.